Hello and welcome to Alexander Disease Research Update, episode number 32, recorded on May 12th, 2026. I'm Albie Messing from the Waisman Center at the University of Wisconsin, and with me today are Dolores Pérez-Sala from the Centro de Investigaciones Biológicas Margarita Salas in Madrid, and Natasha Snider from the University of North Carolina, Chapel Hill. Natasha's been on many times, but Dolores, since this is your first time on the podcast, can you say a bit about yourself and how you became interested in Alexander disease?
Sure, Albi. Thank you very much. And thank you, Natasha, for allowing me to share this conversation with you. It's a real pleasure. So, yes, I've been working in Madrid for several years now with the interest in the post-translational modification of proteins. Modification of proteins not only by inflammatory mediators, but also by reactive agents and even drugs. And these modifications in proteins control their structure and function and allow very fine-tuning order, function, and the coordinations of the cellular events. And during these studies, we came across intermediate filaments and learned that they were really very responsive. They could be very finely tuned by modifications. And so became very interested in pimentin, but also in GFAP. And so we're trying to learn now how these modifications in GFAP control its function. And of course, if they can contribute to pathology. And this is where our interest for Alexander disease started.
Well, thank you again for being here today. So today our topics will be two papers that have just come out of Dolores's lab that address key questions about how GFAP mutations may be changing the basic biology of the cell. So the first paper is by Goya-Iglesias and colleagues and is entitled Alexander Disease Mutations Differentially Sensitize Glial Fibrillary Acidic Protein to Post-Translational Modifications and Network Disruption by Oxidants, which was published in the journal Redox Biology. So first of all, Dolores, what is the main goal of this study? And then tell us how you approached it.
Yeah, well, this is a very long and complicated title for a paper, but in more simple words, what we try to do is to take a further step on the understanding of how the function of GFAP is controlled. And in that field, many labs have done important advances. Even, Natasha's also has made very important contributions to the field of regulation of GFAP by modifications. As you know, GFAP can exist in the wild type form or in different variants. Some of them are pathogenic in different degrees. And our hypothesis was that given the fact that their sequence changes, they could display different susceptibility to damage. Let's say by oxidation, which is our main interest. And so this paper tries to analyze using very, very simple models like the purified proteins themselves in the test tube or cells in which we express the different proteins, the wild type or the variants, how the proteins assemble in cells and how they respond to stress, above all in terms of oxidation. Because when proteins are oxidized, and above all GFAP is very sensitive to oxidation, their assembly can be altered, and that can contribute to the alterations that occur in cells in the disease. So that's our goal and our models.
I think we need to spend a little bit more time talking about what oxidation is and what oxidative stress means for a cell. Those are terms that many people are not completely familiar with.
Sure, yes. Actually, oxidative stress is a process that is very important in pathology, but it's quite simple to understand. It's a matter of balance. You know that all of us live in an oxygen atmosphere, so we breathe oxygen, we use oxygen, our cells need it for many functions, and above all to produce energy. And during that use of oxygen by the cells, some byproducts are generated. And some of these byproducts of oxygen are reactive, meaning that they can bind or attack other cell components, including lipids, proteins, or DNA. So these byproducts of oxygen that are reactive are called reactive oxygen species. And actually, usually they are kept at low levels by the antioxidant defenses of the cells. So we have systems in the cells that keep these active species in non-harmful levels. But under some circumstances, for instance, in some diseases, these reactive species increase to a level where the cell defenses cannot cope with them, and so there comes the disbalance and the oxidative stress. And the problem there is that cell components can be damaged by these reactive species, so the cells become sick.
Well, one of your main focuses in this paper was on a single amino acid in the GFAP protein at position 294. So GFAP in its major form is 432 amino acids long. So pretty much right in the middle is a cysteine at position 294. Can you talk a little bit about why you think that cysteine is so significant, especially in the context of oxidative stress?
Yes, several intermediate filaments display a single cysteine in their sequence. And from former work with vimentin, but also with GFAP, we found out that the cysteine is quite reactive. It means that it can be modified by many species, by not only by oxidation, but also by lipids, and actually acts as a sensor. So as you say, it must be located at a strategic point in the sequence, meaning that its modification is able to elicit changes in the assembly of the whole filament. So we've been very interested in that residue. We think that that is one of the main components of the sense of oxidants by the protein, by GFAP.
And do you also think that this leads to cross-linking of the protein? So two GFAP molecules become bound to each other in a way that is different than the way they normally assemble into a multi-subunit structure. But cross-linking at cysteines would do something different?
Yes, that's right. Because cysteine in the structure of the filaments are close, but they are not meant to cross-link. So they should be forming like a cluster where they are nicely sitting close to each other. But when they cross-link, the distance between them becomes very, very short. And so the structure of the protein is distorted. So it's no good for the filament formation that cysteine residues from different monomers of GFAP are cross-linked. And this, we know that it happens under oxidative stress. And yeah, the functional consequences are quite obvious.
How can we prevent this? Most of us don't really understand what oxidative stress is, but we all hear about antioxidants all the time, especially in the context of aging. What are they and how well do they work? Maybe Natasha, it's your turn.
Okay, it's a field that's been around for a long time. It's just very hard to pin down, when we're talking about oxidative stress, which reactive oxygen species, how local do they act, or how much they can diffuse, how much of it is being produced in the cell. And so a lot of the things that have happened in cells and in animals, unfortunately, haven't translated into human clinical context. So in terms of what has been tried, I think, certain vitamins like vitamin C, vitamin E, for example, if you give high doses in animals, it can rescue some effects, but you cannot give a high enough dose in humans that you're not going to cause toxicity to get enough of it in the brain. And so this antioxidant defense system that's active in a lot of different tissues, it's sort of limited in the brain. And so getting extra enzymes, for example, superoxide dismutase or small molecule antioxidants to the brain could be one potential strategy. And maybe Dolores knows more of the contemporary literature on this. But as far as I know, that it's the delivery, the blood-brain barrier, that's a limiting factor of how much you can get to the brain. But I wanted to bring, I always kind of look at Alexander disease in the context of other intermediate filament diseases and what has been done there. And both of you probably remember Pierre Coulombe's study from 2007, where they found a broccoli compound, sulforaphane, was helpful in epidermolysis bullosa simplex, which is caused by mutations in keratin 5 and keratin 14 genes. These are like GFAP, they're the main intermediate filaments, but in basal keratinocytes. And so interestingly there, with sulforaphane, there was an induction of NRF2. This is a transcription factor that's actually a master regulator of a lot of antioxidant genes. So one way to approach this is not to give, let's say, glutathione itself, which scavenges these reactive oxygen species, but actually to introduce an entire program that could help the cells make more of these glutathione producing enzymes. And interestingly, I kind of followed up on that, and they've taken this to the clinic with some topical application of sulforaphane. Still smaller studies, but it is interesting that some of these intermediate filament diseases that involve similar mechanisms, namely the protein aggregation, could share mechanisms of how the proteins are damaged. And especially for intermediate filaments, I think this is important because they're kind of self-sacrificing proteins. They'll take on the damage themselves to protect other organelles. So in the case of GFAP, where it's in Alexander disease, where itself is mutated and contributing to the damage, then taking on the extra damage, it's probably really amplifying this cascade. So it's worth, I think, investigating different approaches. And one of the things that I had, one of the questions I had when reading Dolores's paper is, I mean, it's always a plus when you compare different stressors. And so you pick 3 different oxidants to compare, and the concentrations and all of that. Obviously, this is an in vitro system, and you were probably trying to max out the response to see the different post-translational sites. But I was wondering if you can kind of relate what was given to the cells to to the cells in vitro. What would the astrocytes experience in vivo, for example?
That's a very good question. Actually, the oxidants were chosen based on previous work by the group of Mike Davis that are experts in protein oxidation. And one of them is hypochlorite that is produced by macrophages, by phagocytic cells in the process of eliminating pathogens. The other one is the result of inflammation. It could be also neuroinflammation because peroxynitrite, that is produced when both nitric oxide and superoxide are produced together. And I don't want to get very specific, but it could be like a product of inflammation. And actually, hydrogen peroxide probably is the most general of all. All of us know hydrogen peroxide. The concentrations are very variable, and there's always this discussion about microenvironments. So how high can go these oxidants can go, and if they are very compartmentalized, then concentrations in the cellular environment can be at some places quite high. So that was the rationale behind those oxidants. Yes.
So sort of the effect was similar across the 3, correct? In terms of the responses.
Actually, it depends on what you look at. The effect is not super impossible because all of them or each one of them induces different mutations. So hydrogen peroxide usually elicits more cysteine oxidation and cross-linking through a kind of mechanism. And the other 2 actually attack other residues in the protein other than cysteine mostly. So you can expect to have slightly different modifications, but in a neuroinflammation situation, you could have the 3 of them. So we just wanted in this article to have like a mechanistic study and get as much information as possible from very distant models. Is not that can be related to pathology, but at least to have a broad scope of the modifications that could occur.
And have you done, or are you doing anything to overcome the oxidative, so adding different types of antioxidants to look at the stoichiometry of how much do you actually need to restore some aspect of the protein folding?
Yeah, well, actually not yet. We would love, and we are planning to do, although it's a lot of work, but we for sure will try some of them. in this article, we tried just N-acetylcysteine. We were asked to do that, and it was quite interesting. As you know, N-acetylcysteine is taken as a drug for cold, just to fluidify the secretions, but it also works as an antioxidant because it's a precursor of glutathione. And we observed there that there was a decrease of the oligomers of GFAP and their control situations, even in the GFAP Alexander disease mutants. Then, when we subjected cells to a strong oxidative stress, this compound was not that efficient, but at least it's something that needs to be explored further. And not only this antioxidant, but also, well, as you said, many others, and trying different strategies, because one possibility is, as you say, to use small molecule antioxidants, but also compounds that activate NRF2, or more recently, I think people are looking more to nanovesicles or exosomes carrying antioxidant molecules in the field of aging as well, that I think they are still very far from the clinic in some cases, but I think it's worth to look at.
The timing of when to give this, and is there a point at which it's not going to do anything?
Sure. And I think I always stress this, the best antioxidant is exercise for all of us. So if we're talking about aging, I think that more than taking any resveratrol or vitamin E or anything, exercise is the best antioxidant because it stimulates your endogenous antioxidant defenses. But in the case of disease, of course, you have to think of something more effective and more realistic than that.
Well, I wanted to talk a little bit more about the cysteine modification, because that's come up a couple of times in the past in publications showing it as a target of oxidation. But you mentioned that your studies have used a cell culture system, and it's not really astrocytes. It's a glioma cell line, which is sort of related to astrocytes, but
That's right.
it's not the real thing. And it's obviously a very convenient model to use, but we need to remember that it is an artificial system. So my biggest question about the cysteine 294 change is whether there's any evidence that it occurs in brain, either in any of the animal models or at best in human tissue.
Yes, it does. Probably not that much in Alexander disease. I've seen
Mm-hmm.
experiments by some of them by Natasha, in which they show GFAP oligomers, but I'm not that sure of how much cysteine residue is involved there. But in other diseases, it's really proven that it's involved in the response to oxidative stress and in modifications, for instance, by 2D electrophoresis or mass spectrometry, the adduct of that cysteine to hydroxynonenal and other reactive compounds has been identified in Down syndrome. So there are several diseases in which it has been confirmed that is a target for oxidation. And in experimental models, probably not sure that it in zebrafish has been shown, but we could take a look at that. So yeah, I'm pretty convinced that it also plays a role in vivo.
Well, it's a piece of information I really hope we can see sometime in the near future to really confirm it for Alexander disease.
Actually, can I ask a question about the working model just to make sure I understand? So the idea is that mutant GFAP in Alexander disease promotes organellar stress, namely mitochondria, which you have shown, and Suchan's lab has shown the mitochondria are certainly altered, and this is where most of the reactive oxygen species would be coming from. So I guess GFAP itself in that context could be promoting oxidative stress. But then once the electron transport chain becomes leaky and you get more ROS production, and if it's, especially if it's contacting the mitochondria, then GFAP itself would become an easy target for being oxidized. And so it's sort of this loop. And the cysteine, the endogenous cysteine that's already there is a prime target for damage. But obviously, if you have a mutant that also introduces a new cysteine, that could become damaged as well. And so do you think testing any kind of antioxidant therapeutic angle makes more sense for mutations that convert some residue to a cysteine, or would it be more broadly applicable regardless of what kind of mutation you have because of this damaging effect that GFAP has on the organelles?
Yeah, well, I think that probably if ever they are proven useful, their usefulness would be broader, not only be constricted to the mutants that have an extra cysteine residue. With the mutants that have the extra cysteine residue, what we see is that these extra cysteines become involved in cross-linking as well. And some of these cross-linkings are really very, very stable, nearly irreversible. So that's not good for the protein. But our surprise of this last article is that we also studied the E373K mutant, and this one only has the 294 cysteine, and this one became highly oxidized and in an irreversible manner. So it's not that the mutant has an extra cysteine or not, it's that the ones that don't have an extra cysteine, maybe because of defects in folding or in assembly, at least the ones that we have studied, they also become more susceptible to oxidation. And these oxidations are not that easy to pick up because they don't form cross-links, so you don't see aggregates, but the cysteine itself is being transformed into irreversible oxidation products. So I think that probably if ever they are being applied, it is probably useful to target them to other variants of the disease, not only the ones that have a extra cysteine residue.
But it does seem like you would have to target the production, the mitochondrial dysfunction first, to reduce the burden, because it does seem like as soon as they're produced, they would affect the protein, regardless of whether you have glutathione around, just because it's so abundant. So it sponges off everything.
I think, yeah, from all the work from many, many groups in this area, it seems that the closer you get to the root of the problem, the more chances you have to get some protective effect.
Well, let's move on to the second paper, which just appeared online this past weekend by Hernández-Gerez and colleagues. And I suppose we should ask you, Dolores, who these first authors are. Are they students in your lab or collaborators, or please tell us.
Well, both of them actually were, one is a predoctoral student and the other one is a postdoc that already left the lab. And we had the great opportunity to hire these people because we got a grant from La Caixa Foundation that we shared with Milos, Beckney, and Eli Ho, and also Mike Davis. And it was a great opportunity to work in Alexander disease. So without that, none of the work could have been done. So Nuria is finishing her PhD now.
Great. All right. So this paper is titled Lysosomal Function, Resistance to Oxidative Stress, and Repair are compromised by expression of the Alexander disease GFAP R239C mutant, which is in press in the journal Free Radical Biology and Medicine. So Dolores, the same questions as before. What was the main goal of this study and what were the main findings?
So again, our main goal is to try to learn a little bit more about the cellular pathogenesis of the disease. And as you know, there were previous evidences on the effect of GFAP, of mutant GFAP on lysosomes. There were several works that were pointed into that direction. We wanted to get a little bit deeper with the tools that we had at hand. So,
but first, let's say a little bit more about what lysosomes are for people.
oh, okay. So lysosomes, actually, you can think of them like the recycling plants of the cell. So they are the organelles where the material that is not needed anymore is degraded, is disposed of. So they have a lot of what they are called proteolytic or lipolytic enzymes that break the molecules the cell doesn't need. And then actually that's very nice because they can be recycled. They can be used in the synthesis of new components. So those are lysosomes. And something that is important about them is that they are contained by membranes, so that these enzymes are quite powerful. They can cut many cellular molecules, are contained in them. They are not split into the cytoplasm. And also they have a quite acidic pH. They are quite acidic in the interior because that is good for the degradation process.
You studied three different variants in this paper, and they had somewhat different effects. Can you explain that a bit?
Well, we actually started the study with three different variants, but we only monitor the distribution and the size lysosomes with the three of them. For most of the paper, we focused on the R239C variant. That was the one that showed the most striking or severe effects.
I see. So this is a more complicated message for me because I don't know quite how well it fits with other things we think about the disease. So it suggests that there would be this generalized impairment of, as you say, recycling, the degradation pathways within the cell. And that's something that's been talked about for a while about GFAP. Maybe one reason GFAP increases is because the degradation pathways are impaired. But there's other evidence that it may be impaired early, but then it seems to restore itself to match the increased synthesis that's going on. So in a steady state setting, maybe there isn't impairment of degradation. And so that's where I'm a little bit confused about where this is going. And that's not the fault of this study. I think it's just the sort of the general state of affairs right now. We don't know what's going on with the degradation pathways.
Yes, actually, this, as you said, this is a cellular model. And what we try to see is whether there's any difference in the behavior of cellular organelles if they express the wild-type GFAP or if they express a GFP variant. So at this point, we are not very concerned about the pathophysiological relevance of the model, because even if we use very distant cells, if the defect is very basic, we think that we will be able to see a defect in very distant cells. So our goal was to compare the two forms of the protein and explore the consequences of the mutation. And of course, we have gone to the one of the most severe mutations, and probably that's why we see this very clear deleterious effect. Maybe with some other variants, we would have seen no defect or not such a strong defect. So, and of course, learning what is really happening with protein degradation in the disease is not something that you can do only with cellular models. You need in vivo studies.
Actually, I have a question. Do we know how much of GFAP normally is degraded through the lysosome pathway versus the proteasome?
We do not.
Someone needs to answer that.
Well, actually, in our cellular model, what we do not see is GFAP, a lot of GFAP inside the lysosomes. So we see the bundles, we see the aggregates, but at least in this cell model, it doesn't seem to accumulate inside. So we probably need to try other models to try to confirm this, but it would tell us that the function of the lysosome is affected, but it doesn't seem to be retaining GFAP.
It's affecting other proteins that have to be shuttled through the lysosome. That's interesting too. It's like, what is the collateral damage and which of the other targets that are affected then make a difference for the astrocyte itself. So you're saying you're doing this in iPSC-derived astrocytes to test that? Because there was in the iPSC model, correct me if I'm wrong, but lysosomes were one of the more affected ones, correct?
In the study that we did with Jeff Jones and Sushan Zhang's lab, that's right. But in that study, we didn't really look at lysosomal function so much.
It was more the distribution
Right.
and the abundance, yeah.
All right, let's sum up very briefly then, Dolores. What would you say are the main take-home messages from these two papers?
Well, I think that you said that at the beginning, that is that we are trying to learn more about the disease, even from simple models that can go from the purified proteins to distant cellular models in which pathogenic variants of GFAP are going to induce alterations. The main message is we're trying to learn and we will continue trying to learn. We hope to be able to have robust models in which we know a little bit more about the pathogenesis. We can also test variants that are not that well known. That is one of our goals. We would like to test one of the less known or not the ones that is not really known how pathogenic they are and compare them to the more pathogenic. And also maybe they give us the opportunity to assay some protective strategies. So I think that's the main take-home message from the two papers.
Natasha, any final comments?
Well, it is interesting, I think, especially if the antisense oligonucleotide therapy against GFAP shows promise and is more widely used in patients to get at least the level of GFAP down, then you can see how layering on these other protective approaches could kind of optimize function at that point. I think the biggest issue is just the amount of protein that we have, but if that's properly dealt with, I think these other types of approaches. So actually, it would be interesting to look at antisense, incomplete suppression by antisense, plus an antioxidant, and whether there's synergy there.
All right, that's all for today's episode of Alexander Disease Research Update. Thanks for listening, and thank you to Dolores and Natasha for joining me today. Our theme song was written by Charlie Allenson, and special thanks to Daniel's Fight, our sponsor for these podcasts. I'm Albie Messing. See you next time.